Hello students, today we are going to discuss on genetically modified food. So these are all the key points that we are going to discuss today. The types of genetically modified foods, the GM food process and techniques, examples of the genetically modified foods, health and safety aspects of the GM foods. How to label the genetically modified foods and what are all the future outlook and conclusion of the genetically modified foods. This is a map of hunger. It is indicating it's a hunger map where it's a map of hunger where it is showing how the people are taking the intake of food material in the worldwide. It cost a little of US 25% a day to food and agri child change your life forever. About 80 million people, one in out of 9 in the world population go to the bed hungry each night. We can achieve the zero hunger in lifetimes. In the world today, we have the knowledge and tools and policies to feed the countries. This map is showing the population of people, those who are with hunger. The green indicates very low and moderately low, and orange indicates moderately high, and red is high, and for brown very high, and we cannot insufficient data for the gray color. So here we can see the many people in the part of the country as well as world, they are having lesser amount of food for the intake. So we are in a position to achieve zero hunger in our lifetime with the help of our technology. Advances in the plant biotechnology have made it possible to identify and modify the genes controlling specific characteristics. Nowadays, scientists can transfer the gene from one organism to the other organism, which is maybe unrelated organism. So, it need not to be from a plant to animal or animal to a plant, producing what is known as genetically modified organism. We call it as, it may be a transgenic plant or animal. Any food that produces this way is called genetically modified food. The foods obtained by added or deleted gene sequence are referred as GMO in short. The genetically modified organism. The food that are coming under the consumption category are called genetically modified food. More technically, the genetically engineered foods are the food that are produced from the plants, animals and microbes that have their genetic code modified by the selective introduction of a specific DNA segments. This process allows the organism to acquire a desirable trait such as pest protection, herbicide resistance, improved nutritional qualities. The enhancement of the desired trait is done by earlier method called breeding. But conventional breeding methods are very time consuming and often will not end up in accurate results. The peculiar feature that we find in the genetically modified foods are the foods that contain an added sequence, which may be from an animal or any other source, or it will have a deleted gene sequence. The animal products from animals fed with genetically modified feed. The products are produced by the GM organism. History of the GM foods. In 1960, a lot of breakthroughs were recorded in the field of genetics. It was proven that this new knowledge has a potential to revolutionize food production. it creates huge benefits for the world. By 1972, another scientific breakthrough was recorded by Paul Berg, who joined together DNA from two different organisms to create first recombinant DNA molecule based upon the Fred Griffith experiment. This breakthrough was by pioneer study in Stanley Cohen and Robert Boyer, who inserted DNA from an African toad into E. coli bacterium. Shortly after this, some companies realized that fledgling technology that will open new and high profitable markets. Some of the revolutionary periods in 1960 to 70, isolation of the restriction enzyme and their use to analyze the DNA structure. This is one of the tools that is used in the production of the GM foods and GMO, genetically modified organism. In 1981 and to 1982, first transgenic animal, or that is a mice, was produced. In 83 to 85, first transgenic plants were produced. In 1990 to 92, first transgenic cereal plant, maize and wheat, is produced. And 1992 to 93, regulation for the deliberate release of the genetically engineered organisms were developed. In 1994, the genetically engineered tomato was marketed in USA. This is the global status for the GM for the foods, the global area of biotech crops. A record of 18 million farmers in 28 countries planted 181.5 million hectares in 2014. A sustained increase of 3 to 4% to 6.3 million hectares, that is 16 million acres over 2013-14. This is the biotech crop countries and mega countries. Growing biotech crops. India is using only lesser amount of the moderate. Here we can see the list of various country which is producing biotech crops, and especially India produces BT cotton. The types of genetically modified foods, we have first generation, then second generation, and so on. The first generation crops, they have been input traits such as herbicide tolerance, pesticide tolerance, and insect tolerance to the environmental stress. These crops are not significantly different from the traditional crops in their appearance, taste, and nutrition. Examples of such crops are herbicide resistant soybean, insect resistant maize, and herbicide and insect resistant potato. These are all the some of the crops which were generated in the first generation crops of GM food. So it is ensuring only pesticide and insecticide tolerance. The second generation crops have new traits to increase the benefit of the consumer, that there is an increased level of proteins in the modified food with healthier fats and modified carbohydrates and increased flavor and micronutrients. Examples of such crops include rice with a higher level of beta carotene, tomato with higher level of carotenoids, maize with increased vitamin C, soybeans with improved amino acid composition, and potatoes with higher calcium content. The third generation crops, these genetically modified foods are in the research pipeline. These plants have the traits that could provide increased ability to resist abiotic stress such as a drought, increased temperature, or saline soils. The other traits that provide health benefits. Yet another objective may create farm plants to produce and achieve pharmaceutical products. In February 2009, the US FDA, that is Food and Drug Administration, approved a license for the recombinant antithrombin for the prevention of blood clots in patients with hereditary antithrombin deficiency. The recombinant antithrombin is the first biological drug derived from milk of goats that have been genetically engineered to produce human antithrombin in the milk. There are three main types of genetically modified food techniques are available, that is inserting gene or called as gene shifting. The genes are determined by different DNA sequences. When the isolated gene is inserted into the plant, it becomes part of the plant and works with its function. This method can increase or improve the plant, such as resistant to insect, where it increases the yield of the food after the insertion. Removing the gene or gene silencing. This function reduces or stops the genetic modification. Example, the function of the virus which causes dried and spotting of tomato is reduced by removing the part of the gene. Thus the virus cannot reproduce, so the tomato can grow healthy. Another way is changing the process of catabolism or gene splicing. The foods can be enhanced by changing the process of catabolism, such as controlling the percentage of starch of the glutinous rice, and it also includes the controlling the taste, mass, color, and usefulness of the food. The process of genetically modified food has 8 steps that begins with how the above said process are achieved means we can isolate the gene of interest, the chromosome which is used to identify the genes that are responsible for the desired trait in the organism. The next is insertion of the gene by using a vector. The desired trait is put into the plasmid. Then plant transformation. The plasmid containing especially TA plasmid is utilized, which is derived from Agrobacterium tumefaciens, that transfers the plasmids and a new gene into the plant chromosome. Selection of the modified plant cells. After insertion, we have to identify the cells which are having our gene of interest. That can be done by selectable marker genes are used to favor the growth of the cells containing the trait as opposed to the non-transformed cells. Regeneration of the whole plant via tissue culture. In this step, we take up the plant, that is explant or cells, into a media containing nutrient that spark the development of the cells to form the plantlet. Once rooted, they are put it into the pot in the controlled environmental condition. Verification of the transformation and characterization of the inserted gene fragment. The tests are conducted in order to determine the number of copies. It does not interfere with the other genes. These tests done to ensure that the gene of interest what we have inserted is found in the modified cells as well as in the tissue culture as well as plantlets which are developed from this tissue culture. Testing of the plant performance. Here we see or test if the resulted plant grown in a greenhouse that has acquired the favored traits and it has any unwanted characteristics. Safety assessment. Now, more tests are carried out to see the plant's performance, environmental safety assessment, and other safety assessments. Here we can see the TA plasmid is taken up from the Agrobacterium tumefaciens, and by using the restriction enzyme, the TA plasmid is cut. In the restricted region, the gene of interest or the foreign DNA is inserted to develop the recombinant TA plasmid. The recombinant TA plasmid is introduced into the cell culture by using a suitable vector. Then after inserting the TA plasmid into the plant chromosome, the tissues are taken up and the cultured for the production. The tissues are taken up, they are cultured, the plantlets were developed, and the plantlets are then potted and it is transferred to the field for the further development. The commonly genetically modified foods are soya bean, corn, sugar beet. It has resistance to glyphosate by inserting herbicide resistant gene, and cottonseed oil by inserting pest resistant Bt crystal protein gene, and tomato by removing the gene that codes for the polygalacturonase responsible for softening of fruits after harvesting. Then potatoes, amylopectin rich varieties by switching off the granule bound starch synthase, thereby it is responsible for the amylose production. Rapeseed or canola with high oleic acid content by adding a new gene. And rice, which is having a high vitamin A that is inserted from daffodils. More specific examples of the genetically modified food includes, first one is golden rice. From the name itself we can identify it is gold in color because of the presence of provitamin A. It was created by Ingo Portricus. The golden rice is a variety of rice produced through the genetic modification to biosynthesize the precursor beta carotene, that is provitamin A, into edible parts of the rice. More than 120 million children in the world suffer from vitamin A deficiency. The golden rice has potential to prevent 1 to 2 million deaths every year which is caused by this vitamin deficiency. The golden rice was created by incorporating the rice with 2 beta carotene biosynthesis gene. One is PSY phytoene synthase and lycopene cyclase. These two enzymes are added to the rice to produce the beta carotene provitamin. So here the geranyl diphosphate is converted to phytoene in the presence of phytoene synthase, which is coded by PSY gene from the mice. Then the phytoene desaturase is converted to zeta carotene. Then zeta carotene desaturase converts it into lycopene. Then lycopene beta cyclase converts it into beta carotene. This is a beta carotene pathway in the golden rice, where we can see the color of the rice. One is in the normal color, another one is in the golden color, hence it is called as golden rice. Next one is cold tolerant tomatoes. The scientists have created frost resistant tomatoes by adding the antifreeze protein, which is derived from cold water fish. The antifreeze gene that is coming from the cold water flounder, the fish that can survive in very cold condition. The flounder has the gene to make chemical antifreeze. This is removed from the antifreeze DNA and is joined into one piece of the DNA called as plasmid, and this hybrid DNA is added to tomato plants to make it a frost resistant tomato. So the flounder fish is taken, in which the flounder chromosome containing the antifreeze genes are then isolated by restriction mechanism, and the TA plasmid is taken and it is restricted with the restriction enzyme, and the antifreeze gene is inserted to produce recombinant DNA. The recombinant TA plasmid is added to the bacterium, and dishes of the bacterium containing the recombinant DNA is selected and it is added to the plant cells. The bacteria, that is the dishes containing the recombinant DNA containing the plant cells, are chosen with the anti-freezer gene of the flounder. Then the plant tissues are then cultured to develop plantlet, and it is spotted to get our GM tomato plant, which is containing the antifreeze gene, which is derived from the flounder. This is the pathway which is showing how we are getting the tomatoes with antifreeze gene. The same manner what we see in the case of the golden rice. Here we are producing vitamin rich tomatoes. The agrobacterium naturally infects the plants by causing the various disease by replacing the genes with the desired one with the gene of interest, which is an advanced trait. So the bright orange color of the tomato comes from the beta carotene, which is the precursor of the vitamin A in our body. So by inserting the color gene into tomato, we can increase the appearance as well as the level of vitamin A in the tomatoes. So how to create a tomato with a carotene? So we take up the carrot and we isolate the beta carotene. The beta carotene gene is inserted into the TA plasmid. The plasmid is incorporated into Agrobacterium tumefaciens. The bacterium is allowed to infect the set of cells which are present with tomato plant tissue. The bacteria infects the tomato plant tissue, that whereby it inserts the gene of interest of beta carotene. Then the plantlets are allowed to grow in a petri dish. Then till they are getting the roots. Then the plantlets are then transferred to the pot for the production of the tomato containing vitamin A, that is the beta carotene, to give more color as well as vitamin rich. Protein enriched potatoes for NASA. Potato is a non-cereal food crop limited in the amount of lysine, tyrosine, methionine and cysteine to provide sufficient protein and amino acids to astronauts with their complete nutritional requirements. A tuber-specific protein, amaranth seed albumin, is introduced to transform the potatoes. The AMA1 protein has a well-balanced amino acid profile In fact, the amino acid composition exceeds the values recommended by WHO for the nutritionally rich protein. This protein was used to do its non-allergenicity in the purified format. When AMA1 gene is introduced in the potato, 2.5 to 4% fourfold increase in the lysine, tyrosine and methionine cysteine content and 35 to 40% increase in total protein was reported in transgenic tubers. This protein rich or the essential amino acid rich potato finds beneficial usage for the astronauts in the space. BT soybean. There are two target insects from the insect resistant and transgenic soybean or the velvet bean caterpillar and soybean looper. So these pests feed on the leaves of the soybean plant that can severely limit the yield of the particular soybean. The velvet bean caterpillar population can reach damaging levels rapidly. Many producers in the area where velvet bean caterpillar is a significant problem, they apply a preventive treatment of dimilin when the plants are in full bloom. Scientists have incorporated Bacillus thuringiensis BT gene, which is derived from the bacterium Bacillus thuringiensis, into soybean, which has insecticidal protein that maintains the yield of the crop. The bacteria produces a toxin called BT toxin. The toxin is capable of killing those pests, that is velvet bean caterpillar and soybean looper. Thereby, it increases the production of soybean plant. The scientists have incorporated the BT gene into soybean, which has been insecticidal protein that maintains the yield of the crop. The Bacillus thuringiensis, the ubiquitous soil bacterium, is the source of the gene for the insect resistant. Genetically modified corn. Corn has been deliberately genetically modified to have agronomically desirable traits. The traits have been engineered into corn, including resistance to herbicide and resistant to pest, insect, and later it has been achieved by the incorporation of the BT toxin, as we seen in the case of the soybean. The corn varieties are resistant to glyphosate resistant, that is a herbicide, have been produced. The pioneer in hybrid has been marked on corn hybrids with tolerance to imidazolin herbicide under the trademark of Clearfield. Here we can see the genetically modified food in the bottom. How it is incorporated into corn means the BT gene is taken up and it is restricted. The BT gene is inserted in the corn expression cassettes. Promoter and terminator are added to it and it is incorporated into BT toxin, this BT plasmid. The vector with the gene is manipulated into the bacterium. The foreign gene is inserted into the corn cell genome and we produce BT corn. Roundup Ready soybean. The Roundup Ready soybean is a transgenic soybean that is immunized with the Roundup herbicide. Since the soybean's natural trypsin inhibitors provide protection against the pest, this is the only major problem in soybean farming was weed that makes your soybean revolutionary. The glyphosate is a herbicide that could inhibit the soybean plant ESPSP gene, which is involved in the maintenance of the biosynthesis of aromatic metabolites that causes the plant to die along with the weeds. For the herbicide was meant a plasmid which was transferred into the soybean cells through the cauliflower mosaic virus to provide immunity to glyphosate containing herbicides. After this process was perfected, the Roundup Ready soybean was ready to fight. First, it was hit in the market during the year 1996. Canola oil. The rapeseed oil has a distinctive taste and disagreeable greenish color due to the presence of chlorophyll. It also contains high concentration of erucic acid. Experiments on animals have pointed to the possibility that the erucic acid consumed in larger quantity can cause heart damage. A known toxin, the cultivar used to produce the commercial food grade canola, which was bred containing only less percent of erucic acid. The levels are not believed to cause harm in the human, and no ill health effect has been associated with the consumption of human with this genetically modified oil. Papaya. Papaya cultivation is threatened by papaya ring spot virus, a disease that sharply lowers the fruit yield. The University of Hawaii developed a ring spot virus disease resistant papaya. To do this, certain viral genes encoding the capsid proteins are transferred to papaya genome. These viral capsid protein elicit something similar to the immune response from the papaya plant. It's like a vaccination program done for the papaya against this ring spot virus. The first resistant papaya varieties were grown commercially in 1999 in Hawaii. These genetically modified papayas were approved for consumption both in US and Canada. The food companies that are using Monsanto products, that is in the alphabetical order they are given here. There are many petitions which boycotts and signs asking them to remove the genetically modified organisms from the food products. Why? The reason is that there are some safety and health issues concerned to the genetically modified food for human consumption. The WHO has identified the following issues that are concerned with the human health with respect to the genetically modified food. First one is allergic properties. Called allergenicity. The GM food that has potential to cause allergic reaction. It is a greater risk of health associated with the traditionally grown foods. However, the proteins produced by any newly introduced gene have the potential to cause additional allergic response. To prevent such allergic properties, transfer the genes from the common allergic food is discouraged unless it can be proven that the protein produced by the introduced gene will not be allergenic. The potential risk in the introduction of the entirely new protein that did not previously exist in the food chain. That is the next problem we come across. Gene transfer. Another potential concern arises from the genetically engineered food is the transfer of genetic material from GM foods to other cells of the human body or bacteria in the intestinal tract will have some problem. So the DNA from the ingested food is not completely degraded because it is unrelated and unpalatable, because the small fragments of DNA from the food found in different parts of the GI tract This will cause horizontal gene transfer to the absorption of DNA fragments to the gut microflora or somatic cells lining the intestinal cell. That may transform the properties of the gut flora, and that may lead to complications of gastrointestinal related disorders. It may even lead to tumor or cancer. The scientists, however, have postulated that the uptake of GM DNA into the cells of GI tract will not have any biological consequences because the DNA will be degraded in the cells. Increase in the anti-nutrients. The anti-nutrient substances that will interfere the utilization of the nutrients. The insertion of the new gene may lead to increase in the existing level of the anti-nutrients. For example, glyphosate resistant Roundup Ready soybean have shown to increase the anti-nutrients. In sheep and cattle, the heat stable anti-nutrients such as phytoestrogens, glucins and phytic acids are found to cause infertility, allergic reaction and decreased availability of phosphorus and zinc respectively. Use of viral DNA in plants. Most GM crops utilizes the cauliflower mosaic virus, the 35S promoter gene to switch on the introduced gene. This is a controversy that whether the cauliflower mosaic virus 35S gene could horizontally transferred to cause the disease, the carcinogenesis or mutagenesis or reactivation of the dormant virus or generation of the new virus into the system. Some scientists believe that the cauliflower mosaic virus found in foods is not infectious and cannot be absorbed by the mammals. Some scientists also point out that humans have been ingesting cauliflower mosaic virus and it is 35S promoter in higher amount, and as it's never caused any disease or recombined with the other virus. Even though many scientists claim that this introduction of modified genes which is derived from the other virus as well as from the bacteria into the gastrointestinal system of the human being, that may interfere the gut flora of the human being. It's not harmful, but there are more possibilities that effect of them on our GI tract is a matter of question. So the testing of the genetically modified foods is made necessitated their impact over the gut flora. The GM products containing additional traits encoded by an introduced gene produces an additional protein that confers the trait of the interest. The raw material or grains and processed products derived from the GM crop must be identified by testing the presence of introduced DNA and their expression by the genetic material. Laboratories carried out assays that are proficient in performing them. How to detect these GMO ingredients? Legislation is enacted worldwide to regulate the presence of genetically modified organisms in crops, foods and ingredients, necessitated the development of reliable and sensitive methods for GMO detection. Protein and DNA based methods employed Western blots, ELISA, lateral flow strips, Southern blots and limiting dilution PCR method have been employed. New approaches are new near infrared spectrophotometry that tackles the problem of detection of non-approved genetically modified food. The efficiency of screening and identification and confirmation strategies needed to be examined with respect to the disappearance of marker genes. Increased use of specific regulator sequence and their increasing number of GM food. In order to identify the normal food from the GM food, the labeling is very, very important. It's an international approach has been carried out to create an awareness among the layman. Internationally, the Codex Amelioratus Commission, an international standard setting body for food, as a committee on food labeling. Since 1990, Codex has sought to develop guidelines for labeling biotech foods. So far, there is no agreement or international standard. In all likelihood, a final Codex standard on the labeling of biotech foods will not occur for many years. The approaches taken in different countries towards GM food labeling differ greatly, as shown in the table below. So, Europe has very strict GM labeling guidelines. In contrast, the United States, Argentina and Canada, three brick producers whose government do not believe in mandatory labeling, Japan, South Korea, China and other countries are in between in Europe and the United States on this issue. The sample of international guidelines for labeling the GM foods. Current labeling policies in the US. The US Food and Drug Administration announces the policy for labeling of GM food. We do not use the ingredients produced using biotechnology. Genetically engineered. The oil is made from soybean that were not genetically engineered. This product contains cornmeal producing biotechnology. High oleic acid soluble is developed using biotechnology to decrease the amount of saturated fat in the soybean. All these are some of the labels that are shown on the products of GM food. Government and GM food. Europe anti-GM protest, and Japan GM testing is mandatory and customer go for organic. In USA, FDA allows the genetically modified foods are substantially equivalent to the natural foods, so no subject to regulation on the genetically modified food, generally recognized as safe. India has no policy yet for the genetically modified food. Below poverty people are unaware of all these things. Brazil. Some states have been banned GM crops smuggled to compete with the grain exporting countries. Africa. Europe opposes the use of genetically modified food in South Africa, Sudan, Zimbabwe as GM laws. Kenya Act as 2009. Argentina very poor pro for the GM. New Zealand no GM food grown here. Current status of the GM food in India. India is a signatory Cartagena Protocol of Biosafety since 2003. India's Apex Biotech Regulatory Committee, the Genetically Engineered Approval Committee, functions as a statutory body under the Environmental Protection Act 1986 of the Ministry of Environment and Forest. Have changed the Genetically Engineered Appraisal Committee in July 22, 2010. BT cotton is the only GM crop grown in India. Cultivation of BT cotton was allowed in 2002. India has become a net exporter of cotton in the last 1 decade. 90% of the total cotton cultivation is covered by BT cotton. The yield is more than 80% when compared to the non-BT cotton. More than 600 hybrid seeds are in circulation now. Here we can see the BT cotton production in India over the years. BT brinjal was placed on a 2-year moratorium because consumer opposed it. So we stopped BT brinjal. We are not the laboratory animals by the Greenpeace movement of the India. There are 55 crops in the pipeline for waiting for the clearance in genetically modified food. The proposed Biotechnology and Regulatory Authority of India called BRAI, the bill will allow the quick clearance of the GM crops by a just handful of people. In India, the labeling is not possible since most of the consumers are below poverty line. The consumer may raise the right of choice if they are aware of the impact of genetically modified foods. In Andhra Pradesh, 3,000 villages with 6 lakh farmers have adopted organic farming, and they said no to pesticide and no to GM crops. The pros of the genetically modified food: it provides improved yield, more resistant to disease, less likely to damage by the insect, tolerance to herbicides, better nutritional value, it has increased shelf life, improvement in the health and environment, better climatic survival by increasing the tolerance to drought, flood, frosty conditions to allow the use of previously inhospitable land, higher crop yields, reduced farm cost and increased farm profit. So the tolerant and resistant crops, pest, disease, drought, frost and flood with increased nutrition with edible vaccines. Where more food, cheaper food when we reduce the risk, reducing the world hunger and improving the world health. Why to produce the GM food? Traditionally, combining the desirable gene in one plant is a tough task and utilizes longer time and needs much attention involving crossing one plant with the another of the same species or related species. From economical and agricultural standpoint, it is advantageous to grow crops that have higher yield or improved quality, pest or disease resistance or tolerance to heat, cold and drought. Desirable genes may provide a mean for plant to combat these conditions. The development of transgenic technology allow us to make use of the useful genes from various living sources to brought together in a relatively simple manner by this mechanism. So here we can see the traits and what are the advantage with the sample products: pest resistance, herbicide resistance in cotton, delayed ripening in tomato, miniature size in watermelon, improved sweetness in sweet peas, cold resistance in strawberries, high starch in potato, and polyester gene is added in cotton to get shining and increase the shelf life, and growth hormone is added in salmon, and hepatitis B virus protein is added in banana. Criticism is there: either mankind will stop Monsanto or Monsanto will stop mankind. This is the controversy of genetically modified food. What are all the criticism or disadvantages of the genetically modified food? Environmental hazard, unintended harm to the organism. It is difficult to design the toxin or kill the crops damaging pest and insect. There is a reduced effectiveness of the pesticide. It develops resistance for DDT. Gene transfer to the non-target species that are crossbreeding. The transfer of the herbicide resistance from crops to wheat. The super weeds will have herbicide tolerance further. What is the solution means? Create a buffer zone. Human health risk, allergic effect is already notified in the case of peanuts. That creates further, it increases the more allergy, unknown effects on human health. And however, the proposal to introduce the genes from Brazil nuts into soybean was abandoned. On the whole, with the exemption of possible allergicness, scientists believe that GM foods do not present a risk to the human health. Economic concern, lengthy and costly process. It may be patented. Monsanto, Novartis, Dow, DuPont hold patents for GM crops and make substantial profit by exporting them. Farmers from the developing countries and economically poor cannot afford. More gap exists between the rich and the poor. Other invention, discourage or stop. Suicide gene technology, only one growing per season. The next time we produce a sterile seed that do not germinate, so we cannot save the seed for future generation. So it is a suicide technology that is developed by this genetically modified organism that put the life of the farmers under risk. Future of the genetic food, modified food. Even though the GM advocates confidence that the next generation of GM food will show even more promising prospects that may also address many of the problems. Australian scientists are adding genes to the banana that will not only provide resistance to Panama disease and a cereal fungal disease that can destroy crops and also increase the level of beta carotene and other nutrients including iron. The GM crops and pipeline including plant engineered to resist drought, high salinity, nitrogen starvation and low temperatures. The current technique that researchers use to introduce the gene into plant cells result in random insertion into the genome. New techniques are being devised to allow the genes to be inserted in the precise location in the genome by avoiding some of the potential unknown effects of the plant genome with the random integration. In future, GM foods will likely to include additional GM animals. That is, a transgenic Atlantic salmon variety is likely to receive marketing approval in near future. Another project, scientists have introduced a DNA sequence into chicken that protects the birds from spreading avian influenza. And although other GM food shows promise for increasing the agricultural productivity and decreasing the disease, this political pressure from anti-GM crisis remains a powerful force. An understanding of the science behind this technology will help us to evaluate the future of the GM foods. Finally, we conclude the session by genetically modified foods are still a new concern in few countries, and its acceptance is restricted mainly due to myth of the consumption, ignoring the valuable benefits. India is the second most populated country. To feed the large hunger and malnourished population is also a challenge. GM crop propagation proves to be the good alternate for the revenue generation in the form of high yield, nutritionally grain with less reliance on pesticides and herbicide, which has no threat to our agriculture and environment. Despite all these positive attributes, this technology still relies in the embryonic stage. Awareness program at village level for farmers to adopt newest technology in the field farming by crop rotation, organic farming and genetic modification results in the production per area with healthy crop. These are all the sum of the references that are associated with this. Thank you.